Hello and welcome to Pharmacy Online. Today I'm going to discuss the process of the cell division. The cell division cycles. Cells reproduce genetically identical copies of themselves by cycles of cell growth and division. So the cell diagram here shows that a cell division consists of 4 stages. G1 is the period after cell division and before the start of DNA replication. Cells grow and monitor their environment to determine whether they should initiate another round of cell division. S is the period of DNA synthesis where the cells replicate their chromosomes. G2 is the period between the end of DNA replication and the start of cell division. Cells check to make sure DNA replication has successfully completed and make any necessary repairs. M is the actual period of cell division consisting of prophase, metaphase, anaphase, telophase, and cytokinesis. As you can see here in this diagram, G1 is the period after the cell division and before the start of DNA replication. S is the period of DNA synthesis. G2 is the period between the end of DNA replication and the start of cell division. Whereas M is the actual period of cell division. Also, cells that stop dividing exit the G1 phase of the cell cycle into a so-called G0 state. What are chromosomes? Chromosomes were first named by cytologists viewing dividing cells through a microscope. A chromosome is a DNA molecule that carries all or part of the hereditary information of an organism. In eukaryotic cells, the DNA is packaged with proteins in the nucleus and varies in structure and appearance at different parts of the cell cycle. Chromosomes condense and become visible by light microscopy as eukaryotic cells enter mitosis or meiosis. During interphase, that is the G1, S and the G2 phase, that is called the interphase, chromosomes are fully or partially decondensed in the form of chromatin that consists of DNA wound around histone proteins. As you can see here, the chromosome, and these are the chromatin fibers, and that consists of DNA that is wound around the histone proteins. In G1, each chromosome is a single chromatid, whereas in G2, after the DNA replication in S phase, as cell enter the mitotic prophase, each chromosome consists of a pair of identical sister chromatids, where each chromatid contains a linear DNA molecule that is identical to the joint sister. So the sister chromatids are joined at their centromeres. A pair of sister chromatids is a single replicated chromosome, a single package of the hereditary information. So now let's discuss what is ploidy. Ploidy is the number of full sets of chromosomes in a cell. Humans are diploid. That means we have 2 copies of each chromosomes. We inherited 1 copy of each chromosome from other mother and 1 copy from each are from our father. So, and also the gametes and sperm cells or the egg cells are haploid, meaning that they have just 1 complete set of chromosomes. Let's discuss the process of mitosis. Mitosis produces 2 daughter cells that are identical genetically to each other and to the parental cell. A diploid cell starts with 2N chromosome that has 2X DNA content. After DNA replication, the cell is genetically diploid, that is 2N chromosome number, but has 4X DNA content because each chromosome has replicated its DNA. Each chromosome now consists of a joint pair of identical sister chromatids. So during mitosis, the sister chromatids separate and go to the opposite ends of the dividing cell. Mitosis ends with 2 identical cells, each with 2N chromosome and 2X DNA content. So all eukaryotic cells replicate via mitosis except the germline cells that undergo meiosis to produce gametes, that is eggs and sperm. So now these are the phases of mitosis. In prophase, chromosomes condense. Each chromosome consists of a pair of identical sister chromatids joined at the centromere. Metaphase, in this phase the chromosomes line up at the middle of the cell along the plane of cell division, pushed and pulled by microtubules of the spindle apparatus. Anaphase consists of when the sister chromatids separate and migrate towards opposite ends of the cell. And in telophase, the chromatids cluster at opposite ends of the cell and begin to decondense. Whereas in cytokinesis, the membrane pinches into and divide the two daughter cells. So here is a simplified diagram illustrating the overall process and the products of mitosis. As you can see here, first the DNA is replicated and then the cells are divided into two diploid cells. Now the meiosis. Meiosis is a type of cell division during which a single cell divides twice and produces four daughter cells. These four daughter cells contains half the amount of genetic material and are known as our sex cells, that is the gametes. Therefore, these four daughter cells are haploid as it contains of half the number of chromosomes of the parent cell. Gametes are sex cells in organisms where sperm are found in males and eggs are found in females. Stages of meiosis. So meiosis cell division takes place in the following stages. Number one is meiosis 1. There is meiosis 1 and meiosis 2. We are discussing meiosis 1 first. Meiosis 1, prophase 1, in which the nuclear envelope disintegrates, chromosomes begin to decondense and spindle fibers appear. Prometaphase 2, in which the spindle fibers attach to the chromosomes at the centromere. Metaphase 1, the homologous chromosomes align at the equatorial plate, ensuring genetic diversity among offspring. Anaphase 1, the homologous chromosomes are pulled towards the opposite poles. Telophase 1, the spindle fibers disappear and the nuclear envelope is reformed. Whereas in cytokinesis 1, the cytoplasm and the cell division result in two non-identical haploid daughter cells. So at the end of meiosis 1, the cells that are formed are two non-identical haploid daughter cells. Meiosis 2. That is, the first phase will be the prophase 2, in which the chromatin condenses into chromosomes, nuclear envelope disintegrates, centrosomes migrate to either poles, and spindle fibers are reformed. Metaphase 2, in which the chromosomes align along the equatorial plate. On the contrary, the chromosomes in metaphase 1 were in homologous pairs. Anaphase 2, sister chromatids are pulled to the opposite poles. Telophase 2, nuclear envelope redevelops and the spindle fibers disappear. And in cytokinesis 2, the cytoplasm and the cell divide, producing four non-identical haploid daughter cells. So here is a simplified diagram illustrating the overall process and the products of meiosis. As you can see, the homologous chromosomes are divided, and then at the end of meiosis 1, two non-identical haploid cells are formed, and whereas at the end of the meiosis 2, four non-identical haploid daughter cells are formed. So what are the main differences between mitosis and meiosis? Number one is mitosis takes place in body cells, whereas meiosis takes place in special cells of sex organs. In mitosis, two daughter cells are formed. In meiosis, four daughter cells are formed. In mitosis, the daughter cells are identical to each other and to the parent cell, whereas in meiosis, the daughter cells are not identical to each other and to the parent cell. The number of chromosomes remains constant in mitosis. The number of chromosomes becomes half in meiosis. The functions include cellular reproduction and general growth and repair of the body, the cells that are undergoing the mitotic cell division. Whereas in meiosis, the functions include producing organisms having different characteristics through sexual reproduction. So if you like my video, please subscribe to my channel for more such videos. Thank you.